2008. And he, I believe, to this day, is still the only professor in our department who actually received his PhD in applied mathematics. So I don't know how many of you guys know that, but Jan is actually a recent convert to biology. So he was an undergrad at MIT, where he graduated with a BS in physics and mathematics. He then went on to get his PhD at the University of Cambridge, as I said, in applied mathematics and theoretical physics. And it was really more as a postdoc that Jan saw the light and realized that studying the cell cycle, and specifically working on yeast cells, was really going to be the most exciting thing that he could do with his future. So he did go on and do his postdoctoral work at the Rockefeller University with both Eric Sigia and Fred Cross. And he is going to tell us about a lot of the exciting studies that have gone on in his lab since 2008, including a couple of recent studies that were published in Molecular Cell and earned him the cover of Molecular Cell. So if you all saw his poster, this is actually on the cover of Molecular Cell. So without further ado, Jan Skotheim is going to tell us about cell cycle dynamics.
Wow. Give me a minute to compose myself here. Right. So thanks for this opportunity to talk here at Stanford. We're quite excited about the opportunity to tell you guys about the things that have been going on in my lab over the last 3 years since coming here. So broadly speaking, what we're interested in is this process of cellular decision making, right? So this decision needs a protagonist. So we start here with a cell, and that cell needs a question. Our favorite question is whether that cell should divide. And to make this decision, the cell bases this decision on a variety of inputs. I've shown here schematically a few inputs. These then get integrated into this decision and then mapped onto some kind of binary choice. For example, shall I divide or shall I not divide? And in the case of cell division, there's specific inputs which people know act through well-known pathways. And these pathways all get integrated at this sort of crucial decision. The cell considers them all together and then comes up with its decision. I shall divide. Right? So that's, broadly speaking, this is how cells make decision. And over the last 30 years or so, we've learned a lot about these different pathways that input into this decision. Right? And so this is sort of summarizing the schematic here, showing our extensive knowledge of the proteins which comprise MAP kinase pathways in yeast. So people have really done a lot of painstaking work to map the components of these pathways and also their biochemical interactions through which these signals get transmitted. However, we know a lot less about how information is transmitted through those pathways. Now, one way to view information transition through these pathways is through input-output relationships. So if we consider, as an example, input, for example, the extracellular pheromone concentration, this will come in handy later in this talk, and that input then gets mapped onto some output, the downstream component of the MAP kinase pathway, for example, the MAP kinase Fuz3. And that relationship can take a variety of different forms. For example, that relationship can be hyperbolic. In this type of relationship, you have a long region where the output is sort of linearly mapped onto the input before it saturates. Another type of relationship, which is well known as an ultrasensitive relationship, where you have this sort of region where the output changes very drastically, even with small changes in the input. And sort of the most dramatic type of relationship you could have is a hysteretic relationship, where, in fact, once you increase your input, you have this sort of bifurcation point, or this sharp threshold, where the system changes very, very rapidly and takes on to another value. And these hysteretic relationships are noted by the kind of bistable regions where you have two different outputs depending on the kind of history of your input. So there's a variety of different input-output relationships that could be a good description of how information is transmitted across these pathways. Now, of course, this is a steady-state picture, and this neglects important dynamics that occur in these processes. And the more sort of general thing really is not to consider just this kind of steady-state picture, but rather signal processing as a dynamic process. So if we consider it this way, we can consider X a kind of vector of concentrations of the important proteins that are involved in that process. And we're going to consider how those proteins respond to some input signal which is a function of time. This can be some arbitrary function of time, right? And the system will evolve, like the concentrations and localizations of the proteins will change depending not only on the current input, but also on the current state of the system, right? So the cell solves these set of equations and gets some concentration field X as a function of time. And your output, of course, is determined by the state of the cell. Vector of concentration is X. And generally speaking, this is going to be some function of all of the different inputs before the time that you're reading it out, right? So like the output is really a function of the history of the input signal in general. So we don't know very much about how cells do this or in what context they do it, but we do have a lot of hope because we have this wonderful thing called microfluidics and monofluorescence microscopy, whereby we can watch cells as they make these decision processes. We can monitor the important proteins that are involved in that process through time, and at the same time control this input signal by our control of the extracellular environment. So our hypothesis is really that it's going to be a great tool to do this, and that they're really going to be this sort of finite set of signal processing functions that are important to cells, right? There's not going to be like you do everything. There's going to be some finite set which the cells find useful, and that we're going to see these types of functions again and again and again. And that these functions are going to be a very useful way of kind of characterizing and categorizing the various signal processing pathways that occur in biological systems. So to be a little bit more concrete, one function which has been found again and again and again over the last decade or so are switches. And these switches are frequently implemented at cellular transition. So a cell goes from sort of one state to another state, one type of cell to another type of cell, not dividing to dividing. And these kinds of transitions are governed by these sort of abrupt transitions, abrupt bistable transitions, not unlike, well, we don't have a toggle switch, but in your house you probably have a light switch, and you can push on that light switch, and you sort of push, 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 then all of a sudden it flips, right? And so this is a concrete example of this kind of toggle switch. The control networks governing cellular transitions are not unlike that. So the regulatory networks, of course, determine these types of dynamics. Typically, what you'll see are sort of arrows connecting proteins, and this is what's called a network topology. And the network topologies are not destiny, because the parameters matter, the strengths of these arrows matter in terms of determining the sort of behavior of the system. However, that said, frequently in these switches, we do find positive feedback loops. It's just an empirical fact that positive feedback is frequently used to generate switch-like transitions and bistability in biological transitions. So there's been a lot of progress in quantitative signaling over the last decade. Kind of some of the buzz of systems biology arises because of this fact. So some functions have emerged with like a whole bunch of examples, like the toggle switch driving cellular transitions. And some other functions are in the process of emerging. Currently, there's like a single or a few examples of these different signal processing functions, but there are likely to be more, right? There are likely to be more of these functions, and the only way we're going to find out what they are is really through this careful study of specific biological circuits to understand how they work, right? That's how we're going to identify more of these. So okay, so far so good. Back to our cellular decision making, to divide or not to divide. That's the question that we're interested in. And this question is answered by the yeast cells in G1 phase of the cell cycle. So this is a bit of quite well-known physiology. It's been known since the early 70s that yeast cells make this all or none decision to irreversibly enter the cell cycle at G1. And the way this was discovered by Lee Hartwell and colleagues was by growing a colony of yeast cells. They're in all different phases of the cell cycle. This is asynchronous growth of a colony of yeast cells. And then at some point, they drop a bunch of mating pheromone on them. And what they find is that some of the cells immediately arrest their cell cycle and engage the mating program, whereas other cells say, I'm already committed to the cell cycle. They ignore the mating pheromone and then go all the way around the cell cycle before dividing again and then initiating the mating arrest, right? So there's sort of dichotomy about pre- and post-start cells, which marks the point of commitment to cell division. So over the last 30 years, people have learned quite a lot about the regulatory network governing G1S transition in yeast cells. And quite briefly, it works the following way. I'm glad Jeremy Thorner isn't here. But anyway, he would probably yell at me. So the upstream cyclin, so the upstream cyclin CLIN3 binds a cyclin-dependent kinase, single cyclin-dependent kinase in yeast, and that initiates the phosphorylation of this protein We5, which is in the nucleus. We5, when it's in the nucleus, is a transcriptional inhibitor. It's inhibiting the SPF transcription factor. This phosphorylation inactivates it, leading to its export from the nucleus, and then weak activation of the SPF transcription. Two targets of SPF are these G1 cyclins, CLIN1 and CLIN2, which are very similar in character to CLIN3, and can complete the inactivation of We5. So We5, when it gets moved to the cytoplasm, is inactive. So that's this positive feedback loop. This interaction here was the subject of a couple of years of my life as a postdoc. And this initiates a very sort of sharp transition into the cell division cycle. Since we're talking about a second cell fate, we have to discuss the second pathway. And so that pathway to consider is this pheromone-induced MAP kinase pathway, or the mating pathway in yeast. And so briefly, the way that works is alpha factor binds a receptor, leading to a bunch of phosphorylation, terminating in the activation of the MAP kinase Fuz3. For our purposes, Fuz3 does two important things. One, it initiates the transcription of a bunch of genes associated with mating. It's sort of the master regulatory factor for that process. Secondly, Fuz3 phosphorylates and activates this factor arrest 1 protein, so FAR1. And as advertised, FAR1 can inhibit the G1 cyclins and stop the cell cycle. Similarly, the G1 cyclins can phosphorylate and FAR1 and target it for degradation, thereby, and also phosphorylate this protein, SERO5, and thereby inactivate the MAP kinase pathway. And if you think about it, this kind of mutual inhibition at the interface between these two pathways is absolutely necessary, because in the one case, you have the mitotic cell cycle pathway trying to make two cells out of one. On the other hand, you have this pheromone-induced MAP kinase pathway trying to make one cell out of two. And you don't really want to be doing these two processes at the same time. So quite a lot has been learned about these two processes. And the question that we got interested in was, well, what can we learn about the dynamics of how this process works? And to look at that, we decided to monitor the transcriptional inhibitor We5 by fusing GFP to it. Right? So this is being expressed from the endogenous locus at the appropriate level. And we can monitor the translocation of this protein, which is a kind of continuous process through G1, which therefore suggests that this would be a very good phase variable to monitor the kinetics of this process. So the first question we had is the commitment point well-defined at all? Like physiologically it's well-defined, you are committed or you're not, but biochemically, is it really well-defined? And the reason that's a question is because it's become, I think at this point, abundantly clear over the last decade of single-cell studies that there's quite a lot of variation in protein concentrations from cell to cell. So what I'm plotting here is the kind of noisomics paper from Jonathan Weissman's lab, where they look at the coefficient of variation in the concentration of every single sort of GFP-tagged ORF in the yeast genome. And what they find is your typical variation in the concentration is somewhere between 10 and 40%. I'll note that these have been gated by size. So you have sort of a typical 10 to 40% variation in the concentration of all these proteins. There are a lot of proteins involved in this decision as to whether or not to enter the mitotic cell cycle. How noisy is this cellular decision, right? You have all this noise and the individual constituent parts. We were wondering if there was a specific molecular event which we could observe that defined cell cycle commitment. And our commitment point assay is quite simple. In fact, it's just a sort of updated version of Lee Hartwell's old experiment, where we grow cells asynchronously in a microfluidic device, and then at some point we expose them to a step increase in mating pheromone. And then we monitor subsequent cell fate. And that data looks like this. Let me turn this down. So here cells are growing and dividing, and you see We5 cycling in and out of the nucleus. Here we add mating pheromone at 300 minutes. The cells then arrest. At 400 minutes, we remove the mating pheromone, and the cells re-enter the cell cycle. And we can monitor the nuclear We5 concentration as a function of time and observe what happens. And what happens is quite dramatic. In fact, we're able to predict what's going to happen by measuring the concentration of a single protein in the yeast cell, the nuclear concentration of the single protein yeast cell. So some cells, which are pre-start, denoted here as pre-start, look as follows. We5 enters the nucleus at the end of anaphase. The cell is divided around here. And then this cell is excluded just a small amount of the We5 from the nucleus prior to the addition of mating pheromone, indicated by the dashed line. The cell then retains the We5 in the nucleus and immediately engages the mating program. So this is a cell that was pre-start at the point of addition of mating pheromone. There's another set of cells that are post-start, right? So these are the committed cells. And these cells look as follows. We5 enters the nucleus again in anaphase, cells divide. This cell has excluded a lot of the We5 from the nucleus at the time that the pheromone gets added, and the cell just doesn't care. You add mating pheromone, it just doesn't care. It excludes the remainder of the We5 from the nucleus and goes through cell division. So you have these two categories of pre- and post-start cells shown here. And we look at about 400 cells and produce a histogram of cell fates as a function of how much We5 gets removed. And so what we can see is that this one measurement is extremely predictive of what's going to happen. If you haven't measured, if you haven't removed a lot of We5 from the nucleus, you're destined to arrest when you add mating pheromone. On the other hand, if you've removed a bunch of the We5 from the nucleus, you're sort of destined to be committed. And as indicated here, there's just one little region of the histogram where you have overlapping cell fates as a function of our measurement. So what does this magical 50% point correspond to? Essentially, if you've removed more than 50%, you're committed. If you haven't, you're not committed. What does it correspond to? And so what we do is we can look at sort of a downstream component in this pathway, which is the CLIN2 promoter, regulates the expression of a unstable GP protein. And so this allows us to see when CLIN2 is being expressed. Now, CLIN2 is a downstream G1 cyclin regulated by We5. We can see this nice periodic expression of CLIN2 as you go through subsequent cell cycles. And as you can see here, there are these very sort of sharp moments where you get this abrupt increase in activity of the CLIN2 promoter. And these abrupt increases in activity in the CLIN2 promoter, as you may have guessed, correspond exactly to like sort of hitting this 50% We5 threshold. So okay. So removal of half the transcriptional inhibitor We5 leads to an abrupt increase in G1 cyclin transcription, which results in activation of this positive feedback loop and subsequent cell cycle commitment. Fair enough. This falls into the category of sort of general category of feedback-driven commitment decisions, but I think we've learned something a bit more than that. Our other question was, related to, given the variation in concentrations of all the constitutive parts, how noisy is this cellular decision, right? Can we measure the commitment point at all? And the answer is, of course, yes, we can measure it. We can get it right to at least 97% accuracy, signifying that this decision, although it has some noisy parts, is not that noisy at all. In fact, it's actually very, very accurate, surprisingly accurate. So on some level, nuclear We5 is destiny. Single measurement results in predicting cell fates of over 97% of G1 cells exposed to mating pheromone. OK, fair enough. So I hope to convince you here that commitment corresponds precisely to activating this positive feedback loop of G1 cyclins. Right? To some extent, commitment corresponds to the sort of abrupt or nonlinear increase in the transcription rate of G1 cyclins, such as CLIN1 and CLIN2. That's here. Now, somewhat surprisingly, these transcription factors, which are at the heart of this positive feedback loop, that's not their sole function. In fact, they have very, very pleiotropic functions, which arise from the fact that they lead to the transcription of somewhere between 5 and 10% of the yeast genome. Right? That's a lot of different genes, which are all cell cycle regulated, and all regulated by our favorite transcription factor at the center of this positive feedback loop. So we thought that was kind of interesting, kind of surprising. And indeed, it's not just that the transcription factor's been chipped there. This is functional. And if you cluster a bunch of these genes, you can see that they cluster together in these kind of somewhat naive clustergram type of images. So our question was, given this apparent complexity of all these things happening at once, could there perhaps be some important, interesting temporal order within the transcription of these SBF-regulated genes. Right? If we look at this a little bit more carefully than clustering everything and throwing it all together, what could we see? And we were motivated by some studies, primarily the earliest ones by Uri Alan, where he argues that in many cases you get this kind of just-in-time transcription where there's an important temporal order, fine-tuned temporal order, ensuring that components of something like a flagella are made just before they're needed in the context of that assembly. So we were motivated by some of these studies. Daphne Kohler is kind of over in computer science has kind of extended this a bit to looking at changes in yeast metabolism. So those were our motivations. And so what we did was the following thing. We took cells, synchronized them in mitosis, and then released them. And we took microarray samples every 5 minutes. Right? And we did this 7 times. So we have 7 different time courses with 5-minute resolution with microarrays. And I'm showing you here the 7 time courses for the CLIN2 gene. And so what you can see here in this experiment, the transcription gets activated here. In this experiment, second experiment, transcription gets activated here. Third experiment there, fourth experiment there, fifth experiment, sixth, seventh. And since we have these 7 data points, we're able to estimate the mean to within less than a minute. So we can estimate when CLIN2 transcription gets activated to within less than a minute using this method. And as we suspected, in fact, the activation times for genes which are known to be regulated by the same transcription factors is in fact different. So perhaps there is some meaning, that at least it's reproducible.
It's causal too.
At least it's, Anyway, so you can see that here CLIN1 is activated very early, CLIN2 a bit after that, and then a bunch of other genes shown here are after that. So now we can look at the timing of the entire SPF-dependent regulon. And we get an interesting result. We find that CLIN1 is in fact the earliest activated gene of all of these, right? And CLIN2 is also quite early, suggesting that these positive feedback elements are activated before the bulk of the cell cycle dependent gene expression. So here's the histogram. We prefer to look at these cumulative probability distributions. The way to read this is that 0.4 here intersects with 16 minutes, meaning that 40% of the genes are activated before 60 minutes into the experiment. Right, so again, we can see CLIN1 is this first blip coming up here. So what does this mean? Well, we think it has some meaning. We think what's going on here is as follows. As the cell cycle progresses, the first thing that happens is that you activate CLIN1. That activates this positive feedback loop, which we've shown corresponds to the decision, exactly to the decision to commit to cell division. And then once you've sort of activated this positive feedback loop, committed yourself to division, you then go through the trouble of initiating the transcription of somewhere between 5 and 10% of your genome, right? So you have this 2-step process whereby commitment precedes the genome-wide changes in gene expression associated with changing the state of the cell. So we thought that was pretty interesting. And so we wondered how general this might be, right? So we wondered, is this just a yeast thing, or is this perhaps more generally applicable? And so what's shown here is the regulatory schematic for the G1S transition of mammalian cells. And there are some similarities. There are also some noteworthy differences. So the similarities include the fact that you have the transcriptional positive feedback loop of these cyclins, cyclin E1 and cyclin E2. Differences include the fact that there's not the only transcriptional positive feedback loops that exist. You also have transcription of this F-box protein, SCIP2, which is instrumental in degrading these cyclin-dependent kinase inhibitors. You also have transcription of this E2F1 transcription factor, which is sort of playing the role of SPF here. The E2F1, 2, 3 transcription factors are playing the role of SPF at the heart of this positive feedback loop. So we wondered if perhaps we could see something similar as what we saw before. Like, is there feedback first regulation in this transition? And so we took a bunch of microarray experiments generated at Stanford a decade ago, unfortunately still the gold standard in mammalian cell cycle synchronized microarrays, but be it as it may. It's on an organism closely related to humans. And in any case, the data is good enough that we are able to see reliable activation of transcription of important components, of the E2F regulated components of the cell cycle. So here's cyclin E1. Here are the 4 different time courses. You can see it's activated here, 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 and here. And we can estimate when the specific genes are activated to within about an hour resolution, compared to sort of an 18-hour cell cycle for these cells. And indeed, just like in the yeast case, we see that a bunch of targets which are known to be E2F targets are activated reliably at different times during the cell cycle, right? So here we have cyclin E1, cyclin E2, E2F1, and a few other targets. And you can see that there are differences in when they get activated. And we can play our same game. We can pool all the 2,000 different known E2F targets together into a distribution and ask, where do our positive feedback genes fall? And quite dramatically, the positive feedback loop genes, shown here in red, fall in the earliest part of that distribution. All of them fall in the earliest part of that distribution, really suggesting that the same thing is going on, right? You have this sort of feedback first motif, which is also being important in regulating the mammalian cell cycle. So to kind of re-emphasize that point, as you're going through this transition, first thing that happens is you crank up this positive feedback loop in green here, decide to enter S phase in an abrupt manner, and only after that you then activate transcription of somewhere between 5 and 10% of your genome, just like in the yeast case, even though these are very different proteins on some level. So from this, we conclude that feedback first regulation is likely conserved across eukaryotic G1S regulation. And I'm going to go a little bit on a limb and make a stronger statement than that. It's like, because of the simple underlying logic, right? It's just commit, then change your gene expression. We hypothesize that this type of regulation is going to appear not only in the cell cycle, but in sort of other important sort of cellular transitions, such as during differentiation. So OK, let's take a step back and go back to my favorite organism, budding yeast. As Martha said, we all have our road to Damascus moments where previous beliefs become untenable. I am Jewish, so I don't know. So, OK. So, right. So these positive feedback loops are frequently found during these transitions, and they act to reinforce specific cellular states, in this case, the mitotic cell cycle, to divide. And in the mitotic cell cycle of budding yeast, it's been shown that this positive feedback loop is instrumental in sort of making this transition irreversible and maintaining this transition in spite of sort of other insults. The other cell fate that we were talking about today was the pheromone-arrested state in yeast, right? So this is a sort of different state. We were wondering if, similarly, this state might be reinforced through some type of molecular mechanism. And unlike in the mitotic cell cycle, you don't have any obvious positive feedback loops in the system that could be performing this function. There aren't any obvious positive feedback loops here. Furthermore, it's a different type of state, whereby the mitotic cell cycle is really sort of an irreversible state where you go into it and you never really go back out of it. You only go out the other end. Pheromone arrest is quite different, right? And there's a very fundamental reason for that. And the fundamental reasoning is that not all attempts at mating are successful. And so my postdoc made this kind of cartoon that I just want to show you, illustrating this point. So we have two mating types in yeast, A and alpha. There's only one alpha cell. As you can see, the numbers are not favorable for the A cells. They chemotrope towards each other. Eventually, this leads to cell fusion of two cells, and somebody is sort of left there. And he could just sort of sit there forever, or he could say, well, this isn't going to work out. I better bud and re-enter the mitotic cell cycle, right? So there's very good reasons why this is not an irreversible state. However, it does take a long period of time. So although it's not irreversible, successful mating requires chemotroping over hours to reach the other cell, and then fusing your cell wall and membranes and so on and so forth. So it does suggest that it would be useful to have some kind of stable state, right? You want to have some sort of relatively stable cell cycle arrest given some set of input signals to perform this process effectively. So our question started out as simply, first of all, is pheromone arrest reinforced over time, right? Simple question, should have a simple answer, yes or no. And the experiment we did was a hysteresis experiment. We took 2 different populations of yeast and exposed them to 2 different histories of mating pheromone, right? So one population followed this blue curve, and they're exposed to this high pulse of mating pheromone for 30 minutes before they're dropped down to the intermediate concentration of mating pheromone. On the other hand, these red cells are only, the red control cells are only exposed to the intermediate concentration of mating pheromone, right? So you have sort of 2 different histories of mating pheromone, and we're going to ask, how long do they stay arrested depending from this point, from time 0, depending on the previous history of exposure to mating pheromone. And so what we find is actually quite dramatic. There's a really significant difference in how stable these arrests are depending on their histories. So if we look at the 6 nanomolar data point here, the cells which experience the pulse are arrested for over 300 minutes. On the other hand, these cells are arrested for just over 100 minutes, meaning that there's sort of a bigger than 200-minute difference depending on these histories, which is significant compared to a 90-minute cell cycle, right? So there's really a very, very big difference here. The cells really care about the history of mating pheromone, not only the current concentration. So okay, so what's this mechanism underlying hysteresis, underlying this sort of reinforcement of the mating arrest? Like, could there potentially be some previously unknown positive feedback elements that we didn't, that just weren't identified before, or perhaps is the MAP kinase pathway slow to reverse its activity? If it's been sort of jammed in the high state, does it get stuck? Does it have a lot of inertia? So to test these 2 hypotheses, well, the first one we didn't test, we just read papers. And 3 different groups have it's good to read papers sometimes. 3 different groups have measured the sort of input-output relationship with the pheromone pathway. And what they've shown is that the system response is linear as a function of the pheromone concentration over about a 25-fold range, right? So the pathway is relatively linear, which is really inconsistent with some kind of unknown positive feedback-based mechanism for reinforcement. We measured that the mating pathway is rapidly reversible. So if you look at something like the downstream transcription rate, or in this case we're looking at it's kind of hard to see, the location of the scaffold protein which mediates the pheromone signal, which exists at the Schmu tip when there's a lot of signal. If you remove the signal, then that rapidly disappears and goes into the cytoplasm in less than 3 minutes. So the pathway is very, very rapidly responding to changes in the extracellular pheromone concentration. So the question remains really, how can this pathway be reversible, rapidly reversible, linear, and at the same time promote arrestability? We've observed all of these 3 things. Something is in conflict here. So to gain some insight into this, we decided to make a model, right? So the MAP kinase Fuz3 activates FAR1 through 2 different ways. One, through the sterol 12 transcription factor, it activates its synthesis. And 2, through direct phosphorylation of the protein, it regulates its activity. So it's activating both the synthesis of the protein and it's activating the activity. This is kind of, in the literature, called feed-forward regulation. There's also some phosphatase which is dephosphorylating FAR1 and inactivating it. We don't know what that is because but I'm sure people are working on it. That leads to this following set of differential equations where we can solve for the active and inactive amounts of FAR1 as a function of the arbitrary history of pheromone concentration alpha of T. It's just a linear system. It's trivial to solve it. But we are helped by the following fact, that phosphorylation is typically, and dephosphorylation are typically much, much, much faster than transcription, and in this case, dilution. So this separation of timescales leads us to have this very nice analytical solution for the amount of active FAR1 as a function of time. And this sort of alpha of T is the alpha factor concentration of the pathway. It's the mating pathway activity as a function of time. And this falls apart into sort of two terms. We have this term here, which is really the fraction of FAR1 which is active. That's determined only by K1 and K3, so that's determined exclusively by the fast phosphorylation and dephosphorylation reactions. And the second term is also very interesting, right? So here we have this nice integral that just screams sort of integrating history and stuff like that. So you have this nice integral, which is the total FAR1, right? So the total amount of FAR1, which is determined by the synthesis rate, is not just determined by the current pathway activity, but is rather determined by the sort of integral of pathway activity over time. And of course, it forgets a little bit because of dilution, right? And this gave us a lot of insight into the problem. First of all, we could see that the fact that phosphorylation, dephosphorylation was very rapid has a lot of interesting properties for this system. First of all, it allows very rapid re-entry even though you have a lot of FAR1, right? So even though you could have a lot of alpha factor over a long period of time, the rest can be rapidly reversed because this prefactor sitting out here essentially responds on the time scale of phosphorylation. So you can very quickly drop the amount of active FAR1 down to 0 and re-enter the cell cycle. The second important fact is that because you have this integral over time, the decision as to whether or not to enter or not enter the cell cycle is not just, the cell is really integrating the pathway activity over time and basing its decision over the, by considering the history of exposure to mating pheromone. So that's a nice model. Our first prediction from this model is that, well, FAR1 should accumulate over time in pheromone-arrested cells, right? Like, start with the simple predictions. We should measure that, and indeed it does, right? So when you expose the cells to a step increase in mating pheromone, you get this nice increase in FAR1 over time. The second prediction is, of course, that when we do these experiments, different cells get different amounts of FAR1 because of this kind of inherent variability of life as a single cell. The prediction is that, well, the amount of FAR1 should correlate with the restoration. And so what we do is we just do the follow the blue curve here, where we expose the cells to 30 minutes of high mating pheromone before reducing it to an intermediate level, 3 nanomolar, and measure two things. First of all, we measure the amount of FAR1 at this point where we drop the cells down to the lower pheromone concentration, and we measure how long they stay to rest. And we correlate these two values, and indeed they're nicely correlated, suggesting that the amount of FAR1 is quite important in terms of determining the restoration. So cells get a variety of different FAR1 levels, and that correlates with how long that they stay arrested, further supporting our model. Now, so far so good, but I mean, crucially, if we say that this hysteresis, the arrest reinforcement, is due to accumulation of FAR1, we should decouple this, right? So we should decouple the FAR1 synthesis from its activity and break this kind of feed-forward motif. And so we can do that. So just to remind you, FUS3 activates sterol 12 with synthesized FAR1. FUS3 also directly activates FAR1. We can rewire this to swap out the FAR1 regulatory region with the GAL1 promoter regulatory region. So now we can control the amount of FAR1 via the carbon source and the activity of FAR1 independently through our control of the extracellular pheromone concentration. And so the experiment looks as following. We grow the cells on galactose for a while and then switch to glucose and measure what happens. And again, we have two different histories of pheromone. One population gets this pulse of high pheromone, and the other population does not. And at time 0, they both are exposed to this 3 nanomolar low intermediate level of mating pheromone. And much to our surprise, this eliminated all of the hysteresis. Not just some, but it eliminated all of it. We would have been happy with some reduction, but for whatever reason, this is all there is to it. And that's shown here. So here's the cumulative probability distribution for how long these cells stay arrested. The blue cells are the ones that get this pulse, and then this dashed red cells are the ones that don't get the pulse. And that distributions look pretty much exactly the same. So that's really all there is to it. Once you determine how much FAR1 the cells have, that's it. That's what is giving the arrest reinforcement. This is just some control cells where you have FAR1 under the wild type promoter under these carbon switched conditions. And as you can see, there's a significant difference between the arrest kinetics. So OK. So I think what we see here is that feed-forward regulation is a nice way to get a rapidly reversible arrest, which is also stable. Like it's sort of rapidly reversible yet stable. And this kind of, without any obvious trade-offs as far as we can see. So although reversibility and stability seem, at first glance to be kind of conflicting things, this is sort of a nice way of doing it where you get both of them without having those two, desired properties, with having those two desired properties. So then one question is, well, why can't this be done by a positive feedback mechanism rather than a feed-forward mechanism? And I think there's sort of an obvious reason for that. So positive feedback, it can give arrest stability, but it's likely going to compromise the sensor, right? So this part here, going from pheromone to FUS3 activity, has this nice linear input-output relationship. This is very good for measuring something, right? Because you have a nice sensor. You can measure different outputs. You have different outputs for a variety of different inputs. This is a good sensor. So feed-forward regulation doesn't compromise the sensor, right? Because you don't have anything feeding back on the sensor. The sensor operates more or less independently. If, on the other hand, you decided to generate stability by throwing some kind of positive feedback loop onto the system, what you'd probably do is convert this nice hyperbolic relationship to something more irreversible or hysteretic, and thereby, because you have this kind of multi-valued output as a function of the input, you've therefore damaged your sensor. And so this is one reason why you would not want to do this kind of thing with a positive feedback loop, but that the feed-forward regulation is superior. In researching, after we kind of discovered this, we went back and decided to read a bit about the literature on feed-forward regulation in cells. And there's quite an extensive, mostly theoretical literature on this concept. There's some original modeling efforts from Uri Alan, and this is reviewed in this relatively good, quite good review from Aviv Regev. But what becomes abundantly clear after sifting through like 300 or so references or whatever, is that almost no experimental work has really been based on this theory. And I don't really understand why that is sociologically, but given the useful properties that we've identified for feed-forward regulation, I don't think it's like unexpected that we're going to see this kind of thing implemented in other cases. Right? It's a very nice way of kind of having a modular kind of sensor and decision-making process. So to summarize, I hope I've convinced you that commitment to cell division is a precise, positive feedback-driven process, and that this kind of that positive feedback activating commitment is induced prior to the other 5 to 10% of the genome, which are regulated by the same transcription factors, via this feed-forward motif. So that ensures that commitment precedes genome-wide changes in transcription. Thirdly here, feedback-first motif has been demonstrated to be conserved in human cells, or at least HeLa cells. And finally, in the last part, I hope to have convinced you that feed-forward regulation could be a good way to get both reversibility and stability with at least limited trade-offs. OK. So I have this is a parenthesis. That was part of the one part of the talk. OK. So this is a curly brace. It should be a parenthesis. And since I have a few minutes, I'll just tell you a few more kind of observations that have been stimulated by this study. So here's a histogram of cell fates as a function of the amount of We5 that's been removed from the nucleus. And it's a remarkably precise process, as I was mentioning before, where you only have this one bin in the histogram where you have overlapping cell fates as a function of the amount of We5 that's been removed. Right? It's so precise that if you use just only this one measurement to predict cell fate, you're going to get it right in over 97% of the cases of G1 cells. Right? So that's quite surprising. And it's particularly surprising because you have this big network of stuff where you have over 20 different parts, which all have some different concentration in the particular cell. And in spite of that, the decision is remarkably robust. So that, in our mind, was really quite surprising. How unnoisy, if that's a word, how accurate the decision was. And that's really, I'd like to emphasize how how that contrasts with other measurements that we and others have made on single cells, where like it's just a kind of constitutive feature of measuring things in single cells is that you see all this variability, right? So this is really the one case where we don't see it. So decision is very robust. There's a kind of a pedantic thing that suggests is, of course, that signaling pathways can be studied independently. Everybody knows this is true. But this is sort of like a new level at which this is true, right? If you couldn't study the pathway independently, or I should say that, yeah, so this is kind of pedantic. You could say, well, what's the alternative to studying signal pathways independently? Well, people do other things, right? So here's an illustration of the yeast interaction network, and each dot is some protein, and each line is like some connection between some protein and some other protein. And when you look at this thing, this is certainly some part of reality, right? There's some part of reality, and it suggests this like interconnected web of everything connected with everything, right? So this is sort of the opposing view of measuring something in one pathway and predicting 97% of. You would never predict that you could predict so well if you look at something like this, right? Because you have your little pathway over here, and then there's this, it's connected to everything. An important thing that we've realized through this process is that we just lack many quantitative definitions. So while this might be a good description for some cellular properties, it's not a good description for others, like committing to cell division, right? So we lack a lot of quantitative definitions. I would say that we have a relatively good one for something like robustness, right? So if we look at some output and we measure the change in that output as a function of change in concentration of some protein, if that change is small, then we say that that process is robust to changes in protein X, right? So in this sense, the commitment decision is robust to changes in many different concentrations in many different components. Where we do worse is in ideas like modularity or complexity. And these are words and concepts that a lot of us use to describe experiments. Complexity is typically experiments that don't work very well. But they're words, and they have some meaning, right? And I think part of it, something that's very complex, is if it's involving lots of different pathways, and you're studying some part of it, but you're not getting a lot of the variation in the output by studying your little piece about it, right? So the other example of like kind of almost total modularity is the pheromone pathway. We're able to predict like all of the output by measuring activity of a single pathway, single protein in a single pathway. Of course, one expects to see things in between. And we need some way of sort of saying how modular or how not modular something is. And I think we do need something kind of like Kolmogorov complexity from information theory. And this is really part of a class of kind of descriptive complexities. And perhaps what would be a useful way of thinking about it in this context is really, how many different pathways are important? How many different protein concentrations do you need to measure to predict what's going to happen in your output, right? So that, on some level, is going to be, how complex is your process? If it's something involving 6 or 8 pathways, you're probably going to need to measure lots of different things to understand what's going to happen. On the other hand, if it's really all determined by one pathway, then one is enough, right? Activity in one pathway might be enough. So I think we need some sort of description like that. And I think this sort of prediction framework, which is, of course, well known to computer scientists and people that do machine learning, but relatively not thought about in the context of cell signaling. I think this is a very nice way of giving you a quantitative answer to how much you understand, right? So if you're forced to predict what's going to happen, you can say, I can predict what happens in 60% of the time, or 70% of the time, or 80%. And it sort of tells you what the residual is. The residual is how much you don't understand about what you're looking at. So I think this is a nice framework for looking at these kind of problems, and I hope that more people start picking it up. So that's the end of that's the end, curly bracket. And we'll just thank the two people that have done the bulk of the work that I've presented here. So this is Andreas Doncic. He's a talented postdoc in the lab. And this is Umut Esser, talented graduate student in the lab from the Applied Physics Department. So I'd also like to acknowledge the other members of the lab. We have a very lively debate, and it's been really helpful for all of us to clarifying our ideas. And of course, funding and people for reagents. And the work I presented appears in these 3 papers. So thank you. Okay, Alyssa.
Clarification question. I but how does that feed forward if the, how does feedback forward if the feedback must happen first before the signal even comes in?
Can you rephrase that question? I'm not quite sure what you mean.
Sure. I didn't understand the definition of feedback forward. And
Okay, no, feedback is if the some downstream part feeds back on some upstream part of the pathway. So the pheromone one is feed forward because the activity, everything is going forward. And so the downstream targets of sterol 12 or whatnot do not feed back to modify the pathway output, defined as, let's say, MAP kinase activity. Does that help?
Yeah.
Okay. So this feed forward is just meant that you don't have feedback. Anyway, Steve.
So I was reminded of this famous quote in your talk, which was that time is just nature's way of keeping everything from happening all at once.
Right.
So apropos of that, the first part of your talk was of course about the establishment of a stable feedback system before downstream targets were activated.
Right.
And that got me thinking about what your thoughts about mechanisms for that. Well, one possibility, of course, is that feedback circuit itself just has fast time constants, so that it equilibrates quickly. But then it occurred to me that same fast time constants could also signal downstream fast.
Right.
So how do you signal that an established feedback loop has been established, even in principle, in order to guarantee that that's upstream temporally from the stuff that happens downstream? Or in other words, have you thought further about how feedback is signaled that it's been established?
Right. So we think that it's not, it's, that what's likely to be going on is that the different promoters, the different regulatory elements, even though it's the same transcription factor, it exists in sort of different parts of the nucleus or in different constellations. And so that makes those factors more or less accessible to the upstream signal. So the thought is that each one of those promoters will have sort of a roughly different threshold at which they get activated. And that has, the CLIN1 promoter has been evolved to be the most activatable of the bunch. So we don't think there's like a direct signal other than that they're responding at different levels of kinase activity. We are investigating the molecular mechanisms that might give rise to these different levels of activity, but we don't have definitive answers yet. Dimitri? Or Charlie?
Feed-forward regulation, to me, suggests that logically, in cell cycle initiation, that you would have some system that senses the availability of all nutrients to permit replication and that this signal would be involved in central activation of cell cycles.
Absolutely, absolutely. The sort of next chapter of what we're going to be doing, hopefully what I'll tell you about in a couple of years, is our investigation of how these different upstream signals impact the activity of the upstream cyclin CLIN3. So it's a big mystery as to, we know that cell size leads to increased activity of CLIN3. We know that nutrients lead to increased or decreased activity of this protein. And we're really very, very interested in disentangling the regulatory mechanisms through which that works. It's a very, very interesting question. Dimitri.
I might have missed this. How does the cell know that it needs to divide by 2 in the first part of the talk? So delta by gamma was 0.5.
Right.
How is that established that it's 0.5 and not something else? And did you vary gamma? Did you vary maximum?
Right. So there's a couple of answers to that question. First of all, we think that it's not a fold response, but rather that it's acting on concentrations of the proteins. You can put in 2 copies of V5 and you get cells that take much, 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 much longer before they enter the cell cycle. So it's much more likely to be a, measuring the concentration. Chemistry works with concentrations typically, right? So that said, we think it's the concentration. We have done experiments, or I should say, Andreas has done a bunch of experiments where we're growing the cells under different nutrient conditions and at different rates and all of this different stuff. And we still get this like 0.5 number, which is really, we don't understand it because we expect it to get, it's just a number. Actually, it's not 0.5, it's 0.48, but whatever. We keep getting 0.48 in one carbon source, another carbon source, another, even though they're growing at almost twofold difference in rate. So this is still a mystery to us. We think there's something interesting here. We don't know what it is yet. But yeah, I mean, maybe there's a really good reason why you want to have it like this, but and it's regulated to be like this in a bunch of different contexts, but we don't really know how or why. Mike?
When you look at that, if I understand the circuitry, right, the SBF concentration, it's rising.
SBF is constant concentration.
Wait, so now I'm confused.
Okay.
If you just draw up the circuit,
Let me
draw up the circuit.
pull it up. Okay.
It's inhibiting SBF activity, or it's inhibiting SBF activity, or SBF?
Activity. So it binds SBF.
Oh, so the question I was going to ask is how abrupt the transition is in SBF activity during this process. In other words, when you finally get the twin 1-2 made, is there a very sudden transition
very,
that then allows?
Yeah. On the transcriptional level, it's almost like a binary thing. And if you model this like a binary, transcriptional activity as a binary process, you can fit these curves very, very well. So it really just, when it goes on, it just starts making stuff at a very high rate very, very quickly. So the SBF activity on a specific promoter changes very abruptly.
And is there anything known about Clin 3, Clin 1, Clin 2, their promoters? Have a few of these promoters been looked at?
Oh yeah, lots and lots of them.
And is there evidence that
It's been bashed extensively.
Is there evidence for multiple binding sites for SBF for the early responders and only single ones?
Right, so we've tried to correlate like multiples of binding sites with early or late responders, and we don't get anything. And we've done a bunch of different correlations. It's not that. Although if you take a specific SBF site and you move it a little bit, you can get changes. Jeremy?
Yeah. Could you speculate on why, or I guess how this system is insulated from the noise like the Wi-Fi system in all the other versions? Like you were talking about the complexity thing. Does that kind of imply that you feel that this system is somehow like not as connected as other systems?
Well, they're all necessary. I mean, every protein is beautiful, right? But they're all necessary. But they're not the variations that exist within the sort of noise within the context of a specific cell might not matter. So they're all identified through screens like inactivating the function, making the yeast sterile, right? Or stopping the entire cell cycle. So they're all really important to have. It just seems to be that this transition is, has evolved in such a way as to be very, very sensitive to particular very few proteins and not so much to other ones. Why that is, I don't know. I mean, it would be quite interesting if this was generally how you want to do it. Certainly you have one very precise interaction which you can very easily change through either the concentration of Wi-Fi or the interface with Clin 3 and tune the system very, very rapidly, evolve it quickly.
You said that you could determine 97% of the cell phase looking at this one parameter. What happens if you look at other parameters or other concentrations of protein in the same pathway? What are the ranges of?
Right. So it's a good question because we just picked one, and you can ask, is that really, special? Is it sort of how special is that one thing that we looked at? Of course, it's very special, but on some level, it's kind of like you're doing this principal component analysis in some 6,000 dimensional space, and I pulled out the x-axis, and I'm like, okay, everything is on the x-axis, right? But you don't know to what extent that is just one of kind of a degenerate set of choices which would give you the same thing. I sort of think that if we could measure some other measurements of pathway activity, we would probably get the same thing. If we had a way to measure SPF activity, you know, like, or phosphostate of SPF, we would probably get the same thing. If we had a way of in vivo measuring the nuclear concentration of CLIN3 plus CLIN2, we would probably get the same thing. So I think there's multiple measurements that would, if we could make them accurate enough, that would give us the same answer.
Have you measured the concentration of CLIN1 and CLIN2 as a fast feedback to put bounds on how strong that positive feedback must be in order to see this cell-fake movement? So not just a transcription level, an actual protein level of cyclin or CLIN2.
We can't measure the proteins themselves because they have half-lives of less than 5 minutes. So it conflicts with the maturation kinetics of even the fastest GFPs. So we would love to measure something like the CLIN3 concentration in the nucleus and the CLIN2 and everything, but there's technical problems prevent us from doing that. Certainly, the switch when you change the transcription rate of CLIN2 is very fast. That's, you know, really, really fast. Okay. Thank you.